Is it on? Yeah, perfect. Okay, so I'm nervous, I have to say. I'm not usually nervous in such occasions, but I am on this one. And I'm very happy to host everyone here for Vesna's PhD seminar. And I'm going to be speaking on behalf of both Govin and myself. Vesna was part of a program that Govin and I have set up for joint research between our two labs at Technion and IBM Research Zurich. Currently, Govin is now at UBC, but over a program, and Vesna did her research basically 50% of her time at Technion, 50% of the time at IBM Research. So this is both a happy day of celebration and also, at least for Govin and me, pretty sad day to really be letting go of one of our most prolific students. And Vesna, I think it's no news to everyone here in the audience. By the way, this is probably the most well-attended seminar, in recent times, at least at Technion standards. Vesna excels simply in everything she does. You'll see it in her presentation, but she made contributions both to basic concepts. You see it in her Nature Chemistry Reviews perspective, to fundamentals, both in separation sciences. You'll see it in her Angewandte Chemie paper, and to fundamentals in physics, in her PRL paper, and to new technologies and applications that are in the papers that are yet to come, or working on them these days. And she's just one of those people that with golden hands that makes everything work. The first year when she joined my lab, I was on sabbatical. I interviewed her before I left on sabbatical, but then she landed without seeing me. She was there for a year before I came back, and we wrote the first paper together without me even being there. So she was independent and an independent researcher from the day she landed. In conferences, first of all, she got selected for all the competitive conferences, twice microtests, MEMS. I can't even keep track. She won all the best talks, best poster awards, and everything she attends. Really, really impossible to follow. And equally important, she's really social glue, and everyone just loves working with her. She gives it all, not only to her projects, but to everyone else's projects as well. And she's also not the glue only within each lab, but also the glue between the different labs. So one leg in Zurich and one in Haifa. And she can talk to everyone in their native language. So pick a language, and somehow she knows it. It's not clear. So no wonder she found herself recruited, being pursued heavily by top labs in the US. I find myself under an attack of emails of people emailing me to try to convince her to go to their lab and not the other lab. So she already made quite a name for herself. And somehow in between, she also finds the time to become an Ironman. Iron woman. I'm not sure what's the correct way of saying that. But for everyone who knows her, she's truly a superwoman. So Vesna, Govin and I are both going to be very proud, are already proud to have been your advisors. We're going to be boasting around, telling everyone you are our students when you're world famous. So just keep using your superpowers to keep doing good in the world. Vesna, stage is yours. Everyone, just enjoy.
Well, Ronnie should have done this at the end, not at the beginning. It's hard to, now I have to manage the expectation of everyone, but I'll do my best. I'll give my thanks at the end of the talk, so if that's okay with you. All right, let's start the slides. All right, do you see my slides?
Yes.
Perfect. Let's turn the pointer. Excellent. So First of all, thank you all for joining. I'm so happy to see you from my undergraduate, my graduate, my PhD studies. I'm really happy to see you here. Today we're going to talk about my PhD work, which is in the topic toward reconfigurable microfluidics, non-uniform electroosmotic flow, and photoactivated high voltage control. And before I begin this presentation, I want to summarize to you what I'm going to talk to you about today, and in fact summarize my entire PhD in one slide. So my work is primarily done in the field of microfluidics, more specifically in reconfigurable microfluidics, which I think is very important and very useful field, and I'll tell you later why. And we looked in one potential way of achieving reconfigurable microfluidics, and that is based on electroosmotic flow, or EOF. But as any research, once we started working on it, we realized we could do different things with this EOF. One direction we took is in the separation of particles and molecules. And while this can be included in some way in reconfigurable microfluidics, this direction has its also side in analytical chemistry. And then we took a completely different direction in cloaking and shielding of microscale objects, and that goes more into the flow physics. And while we had this concept, and we use this concept of reconfigurable microfluidics and EOF to demonstrate two fundamental applications, the common challenge to all this is that EOF requires high voltage. And very quickly we realized that there are no elegant solutions that allow us to control high voltages. So to overcome this challenge, we came up with a new technology based on photoactivation that allows us to do high voltage control. So in my talk, I'll address all these points, but first I want to begin with reconfigurable microfluidics, and maybe even take a step back and see how today a traditional microfluidics is done. So this is a typical microfluidic chip in which we have these physical carved channels, and the geometry of these channels defines the functionality of this device. And the way we fabricate them, we need to design them. Then we're going to fabricate them either using a 3D printer or in the clean room or another approach. And only then we can do our experiment. And if you're doing your experiment and you realize that you want to change that functionality of your device or add additional functionality, you have to go back. You have to redesign, refabricate your device before doing the experiment. So in our lab, we try to break away from traditional microfluidics, and we have a vision of a reconfigurable microfluidics. So in this perspective, we describe this vision of reconfigurable platform in which the user simply draws the desired flow field, let's say on a computer, and then the chip reconfigures itself to produce the desired flow field in real time. We believe this programmability is useful for many applications, especially in the domain of biochemistry and analytical chemistry. For example, we believe such platform would allow chemists that work on synthesis to program and select the desired synthesis protocol and then correct for deviations in real time. Or if you're in single cell analysis, such platform would allow you to select individual cells, and then based on the observed behavior, you can make real-time decisions. Or if you're working in the separations, then such platform would allow you to program it to capture the analyte on demand, and even to couple several separation techniques. So in my PhD, we explored electroosmotic flow to achieve these reconfigurable flows. So to make sure that everyone is on the same page, first I'm going to briefly explain you what EOF is. So if we take a solid surface and we place it in a liquid, most likely this surface will become charged. For example, if you take a glass and place it in a liquid with pH roughly above 4, the glass will become negatively charged. And if we look closely at the solid-liquid interface, we will see a layer of counter ions that are screening this surface charge. And this developed structure here, it's known as the electric double layer, or EDL for short. And the potential at this slip plane is known as zero potential. We can calculate the thickness of the EDL. This is the parameter lambda d, and we see it depends on the thermal energy, but also depends on the property of the liquid. So we have the liquid permittivity and the concentrations of ions we have in our liquid. If we take an electric field now and we apply it parallel to this surface, this electric field will exert a force on this net surface, or net charge in the EDL, and it will create a liquid motion that we call electroosmotic flow, or EOF. We can calculate the profile of this velocity as shown with this equation, and we see that it depends on the liquid properties. So again, we have the permittivity and the viscosity. It depends on the applied electric field, and it depends on the zeta potential of your surface. In microfluidics, very often we are in what we call a thin EDL regime. That means the height of our channel, which is in the order of tens of microns, is much higher than the thickness of this EDL, which is in the order of 1 to 10 nanometers. And in this case, we can drop this exponential, and we see that we are left with this simple equation. If we want to control the EOF field, one parameter we can control is the zeta potential. And in fact, Evgeny from our lab, he developed a theoretical model that gives you the needed zeta potential distribution to obtain a desired flow field. So now that we had the theory in the lab, we asked ourselves how we can control experimentally this zeta potential distribution. And here is our method. We use a phenomenon which we called AC field effect electroosmosis. So we have a microfluidic chip. We have two reservoirs. In one reservoir, we play a ground electrode, in the other one driving electrode. And these two electrodes will allow us to create an electric field inside the chip. On the bottom of the chip, we have what we call a gate electrode. So this electrode is embedded in the surface of the chip and is covered by dielectric. We apply an AC voltage to this gate electrode, here shown in the dashed line. And in sync, we apply a voltage in the driving electrode with this gate voltage. And based on the potential difference between these two voltages, we can locally control the surface charge on top of the gate electrode and therefore locally control the resulting EOF. Here in this video, we just see the flow created by a single gate electrode in the circle. And as you can see, we can not only control the intensity of the EOF, but we also can control the direction of the flow. We can combine several of these gate electrodes, as we see in this video, and we can use it to shape streamlines. And this dynamic streamline shaping, we also applied it for sorting. So you see here we have two gate electrodes, and we use them to sort, in this case, 1 micron beads. But you could very easily imagine such platform could be used to sort, for example, cells. One particular flow field we could create with our method is a bidirectional flow. So as you can see, we have particles moving to the left and particles moving to the right. We call it impossible, not because it's impossible, because you're obviously looking at it right now, but impossible because we cannot create it with other actuation method, for example, with a pressure-driven flow. And this dashed line you're looking at, they're just there to help us to visualize. In reality, this is an open chamber, and within that open chamber, we have this bidirectional flow. We found out that the bidirectional flow could be very useful for separating particles and species. So let's see how that works. For simplicity, we take only two streams. Let's say they have the same flow velocity, and the width of this stream is W. Now let's see what happens if we place some particles. So if we take a high diffusivity particle, this particle will be advected, and we can calculate what is the advection time scale for this particle to be transported by some distance L. But a particle will not only be advected, but it also will diffuse. And we can calculate what is the diffusion time scale for this particle to diffuse across this stream. And when these two times are approximately the same, we define this length as a penetration length. And this length basically tells us what's the maximum length that the particle will penetrate into the channel before enters a stream with opposite direction and basically is brought back. We see that the penetration length depends on the flow configuration, but also depends on the diffusion coefficient of the particle. So now if we take a lower diffusivity particle, this particle will be mainly advected, which will diffuse less, and therefore it's going to be separated from the green particle. We call this separation method bidirectional flow filter, or BFF for short. Bidirectional flow, I think it's obvious, and filter because we begin with a collection of particles and we retrieve only some particles. So it acts as a filter. But we're not using any membrane or centrifuge, but we're using bidirectional flows. So let's see how this works in experiments. So we are looking at the high diffusivity dye. We see that it enters the channels, but then essentially stops. That's because it reaches its penetration length. But when we add beads, these 1 micron beads, they have much lower diffusivity than the dye. They remain on these streams and they're advected downstream and therefore separated. This is just an example that serves as a proof of concept of the separation. We used our method to do something more useful and to separate labeled antibodies from labeling dye. So we begin the experiment by mixing these 2 species in the inlet, and then we start the separation. This is what we observe near the inlet. So the red signal you see, that's the fluorescent signal coming from the dye. We see this dispersed front. And the white is the fluorescent signal coming from the antibodies. So near the inlet, we see the clear separation between these 2 species. And near the outlet, we see that only the dye reaches it. And for a sanity check, we can plot the intensity profiles across the channel. And in red, we have the signal for the dye, and we see that the dye penetrates up to 1 millimeter, which corresponds to its penetration length. Whereas the antibodies, they reach all the way to the outlet. We do lose some of them, but at the end, we end up with a pure antibodies without dye. Now that we have we saw the separation concept and we saw some experimental demonstration, it's very useful to know how we can tune the separation. If you want to design a separation, how you can tune the parameters of the separation. So essentially our parameter that we can tune is the penetration length. So we saw this penetration length tells us how much the species penetrate into the channel. And we see that this penetration length depends on U, the flow velocity, the width of the stream, and the diffusion coefficient. So if we increase U, the penetration length increases. Why is that? So if flow velocity now is much higher, the particles for the same time, they're going to be advected by much larger distance. That's why the penetration length increases. Or let's now say if we decrease the W, we decrease the width, we see that the penetration length decreases. And that's because if we decrease now the width, the particles will need much less time to diffuse across the stream, and therefore they will be advected by a shorter distance. That's why the penetration length decreases. And even though this is a scaling parameter, we also observe it in the experiments. For example, in this video, we're looking at the fluorescent dye and we're tracking its front. And we see that it enters the channels, but then essentially stops. So the video is still running, we're still tracking, but the dye is not penetrating further into the channel. And the reason is that the dye reached its penetration length and is now no longer going inside the channel. We validated this scaling for different flow configurations. So here we're looking at the penetration length as a function of this parameter, this scaling argument. And we tested different widths and different flow velocities, and we see that we have a linear one curve, so we have a linear agreement between these 2. So now if we want to design the separation, all we need to do is to design the penetration length of both species such that the penetration length of the low diffusivity of the ones we're interested in should be higher than the length of the channel. So we want them to reach all the way to the outlet. And the penetration length of the high diffusivity species, the one that we don't want to retrieve after a separation, we design their penetration length to be smaller than the length of the channel. And that means they're going to remain within the channel and they will not reach the outlet. For a more complete model, we solve for the advection and the diffusion for both species. I'm not going to run you through the model. I'm just going to show you the takeaway message from this model. And one important parameter for people in separation is the purity of a given separation. So we define the purity as the concentration of the species that we have in the outlet that we're interested in, divided by the concentration of all the species that we have at the outlet. Ideally, we want 100% purity. We don't want any species that we're not interested in reaching the outlet. So in this color map, I'm showing you the purity. And we have on one axis the diffusion coefficient of one species and the other one diffusion coefficients of the other species. And these are the flow configuration we used for this example. Let's say we want to separate dye from antibodies. So we're tracking the antibody diffusion coefficient and the diffusion coefficient of the dye, and where these two curves intersect, that's the expected purity. We see we have very high purity, and that's because the dye and antibody have very high difference in their diffusion coefficients. Similarly, if we want to separate cells or DNA, we also have very high purity. But let's say we want to separate antibodies from proteins, then we're in this part here, and the purity is not so great. It's barely above 50%. But remember, we said the penetration length is our tuning parameter, and basically we can tune U and W to improve that. That's what I'm showing you in the next graph. So this is the purity now for the separating of proteins and antibody as function of the width and the flow velocity of the bidirectional flow. So if in this case, we were here, we were at 100 micron width and 40 micrometers per second, so these conditions, we can simply decrease the width to 20 microns and therefore increase significantly the purity. So now that we know how, what's the theory behind the BFF, you can design for your own species, you can design the flow configuration to achieve the purity you would want to. But how about extraction? So far, I was talking about separation, but very often we want to have access to the separated species for further analysis. And in fact, with our design, what we call the separation design, the extraction is not possible. And here's the reason why. So at the outlet, due to mass conservation, the streamlines must recirculate, and therefore they carry back the separated species back in the channel. So in the second part of this work, we redesigned this geometry, allowing for extraction. So we took the outlet and we split it into two outlets. So we created the Y junctions. And now when the species reach the end of the channel, now they're directed to this top reservoir from where they can be collected. And to minimize any pressure imbalance between these two reservoirs, we designed what we call an equilibration channel that minimizes this pressure difference. All right, so now let's see how the extraction design in action. So we do the very simple example as before. We take 1 micron beads and dye. And first we look what's happening near the inlet. So near the inlet, we clearly see the separation. So this bright signal is coming from the dye, and the beads are remaining on this stream that carries them towards the end of the channel. And if we look next to the Y junction, we indeed see that the beads, now they are redirected and they go this top reservoir from where they can be collected. We also showed a separation of the DNA molecules with different lengths, but for sake of time, I will not go into this. I'm not going to show you these results. And if you're interested, you can look at our latest paper in analytical chemistry that is specifically on that. So to finish with the BFF, just to give you an idea of what the BFF could be useful for. So the chip implementation of the BFF makes it very useful for low samples. And low samples are found in single cell analysis or when you are dealing with rare or expensive samples. And current sample preparation methods are not suitable for low sample volumes. And therefore, we see the BFF as a sample preparation method for low samples that would enable further downstream analysis. All right, let's go back to our summary slide just to see where we are. So we saw the reconfigurable microfluidics concept. We saw how EUF is a one way of achieving that. And we took this path towards analytical chemistry with the BFF, and we saw how we can separate molecules and particles. But now let's take this parallel path to it and let's go into flow physics. So using the same EUF that we use for the separation, we also show that we can do hydrodynamic cloaking and shielding. But what is hydrodynamic cloaking and shielding? So when we think about cloaking, very often we think about the optics world. For example, if I take Harry Potter's invisible cloak and I wrap myself, I will become invisible. But in a way, it means that the electromagnetic field, which is light, is unaffected by my presence. So very similar analogy, hydrodynamic cloaking is when the flow is not affected by the object. So this schematics shows it very nicely, this concept. So this is the flow, how the flow would look like without an object. The moment we put some object, the flow is affected. And if we want to cloak the object, we basically want to create this configuration here where the flow outside some certain region is not affected by the object. On the other hand, hydrodynamic shielding is when the object now sitting in this chamber here does not feel any hydrodynamic forces. So far, hydrodynamic cloaking and shielding has been shown using metamaterials. So this is a static approach. You cannot turn on and off your cloak and your shield, and you cannot switch between two states. In collaboration with Professor Hart at Darmstadt and his team, we showed that we can do dynamic hydrodynamic cloaking and shielding. And here's our approach. So our object is sitting in a microfluidic chamber, and around the object is what we call a cloaking shielding region. So let's look at the cloaking conditions first. So cloaking means that outside of this cloaking region, the flow is not affected by the object, and we see this straight line. Now the color map here shows us the pressure. So if we want to have shielding, we want to have a uniform pressure around the object, because uniform pressure means there is no hydrodynamic forces on the object. And to create these conditions of cloaking and shielding, we use a gate electrode, the very same gate electrode we use to create the bidirectional flow. And we use that to achieve the cloaking and shielding conditions and switch between them. The question now is, how do we know which flow field is required to obtain cloaking or shielding? And this is where the theory comes into play. So here are our governing equations of the system. This is the equations for the flow velocity, the equations for the pressure. And this U-slip is basically the flow field created by our gate electrode. If we want to have hydrodynamic shielding, we said we don't want any hydrodynamic forces on the body. So what we do is we calculate those forces. We basically integrate the pressure and we set them to 0. And we plug this into our governing equation, and we solve for the flow velocity created by the gate electrode. On the other hand, if we want to have hydrodynamic cloaking, we said we want the flow field to not be affected by the object. So outside of this cloaking region, we set the flow field to be as if there was no object. So again, we take these conditions, we plug it back, and we can solve for this U-slip. So here is a comparison between the theory on the top and the experiments on the bottom. And the color map in the theory is it's the pressure in the system. So if we have just a pressure-driven flow, we see that in both in theory and experiments, the streamlines are bended around the object. In the shielding conditions from the theory, we see that the pressure here is uniform in the same color. And in white, we see the corresponding streamlines, which we also observe in experiments. And in cloaking, outside of this region, we see the streamlines are straight, they're not affected by the object, and we see the very same behavior in the experiments. And unlike traditional methods that use metamaterials and are static, the main point here is that we can switch between states. So for example, here we have a pressure-driven flow, and then we can turn on the shielding mode, as you can see. And from shielding mode, we can now turn into the cloaking mode, as you can see now, and now the object is being cloaked. So now that we saw these two branches, we saw how UF can be used for two different things. Let's go back to our initial idea of using EOF as a method to achieve a reconfigurable microfluidics. So let's say this is a single gate electrode sitting in a microfluidic channel. We saw that this single gate electrode can create this flow, these dipole flows. But if we want to get closer to a reconfigurable microfluidics, we want to create a variety of flow fields. And for that, we would need an array of gate electrodes. We would need many of these electrodes. And while conceptually seems very simple, the problem with this gate electrode is that they require high voltage. They require about 100 volts. And while for low voltages, multiplexing solutions are available. For example, your screen you're looking at is a multiplex solution. You have a CMOS chip that tells which pixel to input what. For high voltage, there are no commercially available solutions. So to overcome this, we proposed a new technology that uses photo actuation and photo switches to achieve a high voltage control. So what is a photo switch? A photo switch is nothing else but then two metal pads, and on top we have a photo material. this photomaterial changes its conductance when it's exposed to light. Let's look at the current that goes through this photomaterial. So when the light is off, we see that the current is very low. That means the conductance of the material is very, very low. But the moment we turn on the light, the current jumps drastically. And that also indicates that the conductance of the material increases. So in order to control individual gate electrodes, we connect each gate electrode with one photo switch. If we don't want to have an EUF generated by that gate electrode, we just don't shine light on this photo switch. Its conductance remains very low, and therefore the electrode in some way it's disconnected from the power line. There's no voltage going onto the gate electrode. If you want to turn on the gate electrode and generate this EUF, you shine light on this EUF, on this photo switch, its conductance increases and basically bridges the power supply with the gate electrode. We could very easily extend this to a chip level. So now we can use a light projection system, which is inherently low voltage, has a high resolution, is commercially available, and it's cheap. So using this light projection system, we can project and select the desired gate electrodes. And important, we use only one power supply to provide the voltages to all these gate electrodes. So for that, we need photoconductive materials. So we explored two materials, amorphous silicon and zinc oxide, and we developed fabrication recipes to allow for the deposition of these materials. Here is just a photograph of such a photo switch. The square pads are metal spaced by a gap, and then on top we put our photomaterial. Using such photo switches, we tested their photoresponse. By photoresponse, we mean what is the ratio between the conductance in the light state when there is light and the conductance in the dark state. For both materials, we have amorphous silicon here and zinc oxide. And the different colors are the different colors of the light we shined on these materials. So amorphous silicon, if you look at its semiconductor structure, has absorbance peak in red. And that's why its photoresponse, it's the highest in red, but also responds well to other colors, as we can see in the green, in the blue, and in the violet. On the contrary, the zinc oxide has absorbance peak in the UV. That's why this response is most expressed in the UV, and it's less sensitive to other colors such as green and red. And now that we have our photoconductive materials, let's see how we can control gate electrode with our photo switches. So here's a photograph of a typical chip. We can see we can fabricate the switches directly on the chip itself. And inside we have the fluidic chamber and we have an electrode array. So just for a proof of concept, we fabricate a 3x3, so we have 9 gate electrode. And here are the flow fields we can achieve. So here in this first video, we can see we can select single gate electrode. So the yellow square is where actually light is shining on, and that electrode is on. We can also select several gate electrodes. We can have this vertical, horizontal line. But what is more important is that we can not only select the gate electrode, but we can set the direction of the flow on that gate electrode. For example, the blue one, for example, the red one goes from top to bottom, and this one goes from in the opposite direction. Maybe it's more visible in this video where we have this line going from bottom to top, and in blue one, we change the direction. And while working on this photo actuation technology, we realized that there are other fields that are also seeking for a multiplexed high voltage solutions. And in particular, we are very interested in actuators because these actuators could be used for a reconfigurable microfluidics. I'll tell you later how. And one of the leading labs working on the field of actuators is Professor Sherry TFL. So they developed these haxels. These are actuators composed of this elastic, the blue is elastic membrane. They fill it with a dielectric oil. And when they apply a voltage between these two electrodes shown in green, they get a snapping action. And this snapping action creates very large forces. As you can see, also very large displacements. And they also have the same problem with us. These actuators require high voltage, even higher than the gate electrodes. They require 1 kilovolt and above. So in a collaboration with them, we try to adapt our photo actuation method to control an array of these actuators. And it's very similar schematics as the gate electrode. We use a light projection system that shines light onto the actuator array, which is integrated with the photo switches. If you look closely now at one actuator, we see that we have a little bit different architecture than the gate electrode. So we have now not one, but we have two photo switches per actuator. If we want to turn off this actuator, we don't shine light on this photo switch. Therefore, we are limiting the current that passes through. But since we always have this dark leaking current and we don't want it to go into the actuator, we shine light on this photo switch here, and therefore the light goes into this branch. Sorry, the current goes into this branch because it has higher conductance. Now, if we want to turn the actuator on, we do the opposite. So we shine light on this photo switch. Therefore, we allow the current to go through. But now we don't want to lose any current in this branch, and therefore we did not shine light on this photo switch here, and this branch has now much lower conductance than the actuator. This could be easily implemented for an array. And again, the important thing here is that we use a single power supply to provide the voltages on these actuators. And here is a demonstration. So we are looking at 5 by 5 actuators. And we can see we can individually address them. This shows you in more details the snapping effect. So we can go one by one and select all the actuators. We also wanted to show something more visual and maybe a fun way to see the actions of these actuators. So we placed a membrane on top of them, and then we put a little ball. And as you can see, we can move the ball around it. So where you see this red square that's where the actuator is off. So we have this off state, and that's why the ball is rolling into there. Of course, our vision is not to move balls, but we see the actuators as a potential way of achieving a reconfigurable microfluidics. So we're thinking of having an array of these actuators and having a membrane on top. And by turning them on and off, we can create microfluidic channels. So now this comes back to my initial idea of my PhD to create a reconfigurable microfluidics. So while in my PhD, I was mostly exploring gate electrodes with which I can control the flow fields. Here is another approach how we can make reconfigurable microfluidics with actuators. And with this, you actually do not control the flow fields, but you change directly the shape of your microfluidic chip. So this is the end of my PhD work and my presentation. But before we open the floor for discussion, I just wanted to tell you what I plan to do next in case Moran and Govin let me graduate. So I decided for my postdoc to do something completely different. And the main reason for that is this. So this is a current state of my plants. And since I'm very bad at taking care of plants, I decided to develop new technologies and new tools and new models to study plants. And in particular, I'm interested in studying the vascular transport in plants. So that's the transport of all the sugars that are being synthesized in the leaves to all the tissues of the plant. And for that, I'll be joining Professor Abe Strook and Cornell, with whom we're going to pursue this work. And now I come to my I think the most important slide of this talk. And this is my thanks to all these people. As you can see, there are many people involved in my PhD that made my journey very, very enjoyable. So first of all, I have to thank my advisors, Moran and Govin. I'm so happy and so lucky to have them. I don't want to brag, but I think they are the best advisors a PhD student could ask for, because they allow you to be independent, and yet they guide you whenever that is needed. And due to their collaboration, I was also lucky enough to be 50% of my time of my PhD in Israel at Technion and the rest of it in IBM, and then to also have two lab members with whom we shared a lot of fun. And so I highlighted some of the names, but I think I'm personally gonna thank all of you, but this is just for you to know all the people who've been involved. And want to thank also our collaborators. We had very fun working with Professor Stefan Hard and his team and Professor Herb at EPFL. And lastly, I must thank the funding agency, because these are the people that pay for our research. So with this, I think we can open the floor for questions.
Thanks very much, Vesna.
Well done. Congratulations.
I'm not done. I'm not PhD yet, so but thank you.
Vesna, congratulations, really amazing. Great talk, you know, through tour de force through your entire PhD and the different topics. And like we said, and thanks for the kind words. We'll open the floor for questions. Isal, maybe turn off the recording so people can feel more comfortable. And I said